Okay. All right. Well, good morning, everybody. Let's get started. We'll be talking about the bidirectional communication between the immune system and the CNS. This is part two of the lecture. We didn't quite finish from last week, so we'll just have a few more slides. And I think, if I'm not mistaken, we ended with this one, and I'm not going to go through all this again. But basically, this is trying to show you kind of that quick signaling pathways with the vagus and with the blood-brain barrier interface. We actually did a paper for our journal club yesterday that talked about this important blood-brain barrier interface and how fast it gets activated. Um, then the perivascular macrophages, propagation to the parenchymal microglia, and ultimately sort of on the neuronal target. So I think this is an important slide to know and to think about. What are some of these key signals? So the ones that we know about for immune to brain signaling, the ones that come up most often are IL-1, prostaglandins, and nitric oxide. And so for IL-1 is interleukin-1. I know we've talked about it. It has to be kind of propagated. IL-1, there's IL-1 receptors on microglia and astrocytes. We think that the astrocytes have an important role in kind of helping microglia. And in this diagram, it's showing the release of ATP, which can bind to prunergic receptors on microglia. IL-1 is made as a pre-pro mRNA. What that means is when it gets translated into protein, it's sort of in an inactive form, and it needs to be processed. And then there's this the cell, in order to actually process IL-1, needs to have something called caspase-1. When I was in grad school, it was called ICE, IL-1 converting enzyme, and then it got changed to these caspases. Caspases are a family of these proteases that can chop things up. Caspases you also hear about quite a bit in that programmed cell death pathway. But in this case, caspase-1 helps process IL-1 and also IL-18. And you see that now with that processing, it chops off the pro form. And you get this active IL-1 protein. As we've talked about, there's IL-1 receptors on neurons. Also, we know that these pathways, the propagation within the brain also involves cyclooxygenase. Cyclooxygenase is an enzyme that converts arachidonic acid to prostaglandins. There's a lot of different types of prostaglandins. A lot of different cells express these. The COX-2 is the inducible form. I think most cells have the COX-1 enzyme. COX-2, I think you can find in myeloid cells, but also in endothelial cells. And prostaglandin E2 is the one that seems to really be neuroactive. There's two types of receptors on neurons, the EP1 and EP2. And these are inside the cytosol. So the PG goes through the cell because it's a lipid mediator, goes right through the cell and binds within the cytoplasm. And again, they'll bind and a nuclear localization tag will be revealed and they'll go into the nucleus. Nitric oxide is a gas. So it's produced by nitric oxide synthase. It's another fast-acting signaling molecule that neurons respond to. So probably all these pathways are involved in propagating the signal. And of course, there's multiple cells involved: microglia, astrocytes, endothelial cells. So this is showing you the connection with interleukin-1 and prostaglandins. This is some of the first finding some sort of classic studies here in the late 90s, where when you added IL-1, I believe this is IP, you add IL-1, you see increased COX-2 expression in the brain. And so what they're doing here is giving these injections and then looking for FOS. So FOS is going to be neuronal activation, and you see it sort of increasing, and then you see neurons getting activated. And they're doing it in the concentration of indomethacin, which is a COX-2 inhibitor. So probably if you guys don't know, a lot of your pain and inflammation medicines that you take are COX inhibitors, so that they block cyclooxygenase and prostaglandins. It's also really good, fever reducer. So this indomethacin, this prostaglandin inhibitor, block this IL-1 induction of neuron activation in the hypothalamus. CRF is cortical releasing hormone. It's something that's going to be activated in these neurons. It's increased by IL-1, and it's attenuated by indomethacin. So here they're giving intramedullary injections of, let's see, prostaglandins, and they're seeing that you increase neuronal activity. So what the model is, and what the model has been for a long time, is what IL-1 signals through partly is this cyclooxygenase, the production of prostaglandin E2. I mentioned these receptors before. And then within the hypothalamus, this is part of what regulates body temperature in the hypothalamus. So a really cool connection that, series of studies showed this IL-1 to body temperature change through prostaglandins. Now the IL-1 signal transduction, I think we've seen it before, at least in the NF-kappa B, or maybe I showed you the TOL4 signaling. But as I told you, that there's a lot of related intercellular compounds. And for this class, one of the main transcription factors of these inflammatory signals is NF-kappa B. I told you about how it gets the I-kappa B, the inhibitor of kappa B, gets phosphorylated by the NF-kappa B activating kinase or initiating kinase, and then it gets degraded by the ubiquitosome or by the proteasome, leaving the active thing. P55 or P65, P50, excuse me, goes to the nucleus, binds, and we see a lot of these inflammatory genes, but also a number of anti-inflammatory genes. So as you're producing IL-1 and IL-6, TNF-alpha and IL-10, you're also producing IL-1 receptor antagonist and I-kappa B, which comes back out and tries to bind up this transcription factor. So again, be familiar with this pathway. At least let's put it this way. If I'm asking you questions about inflammatory signaling, and I'm asking, don't pick protein kinase A or, a different signaling pathway. If you're going to guess for NF-kappa B, right? Okay. So why do we have this propagation within the brain? Why is this necessary? Why can't just the cytokines that, bind to the endothelial cells control everything? Well, the brain is under that tight anti-inflammatory control. Cytokine mRNA is very unstable and has a short half-life, meaning that it doesn't stay along for very long. So it's kind of a very limited amount of production. So you don't see a lot of it, and it gets transcribed and then translated, and then it's gone. Cytokine action is either paracrine or autocrine. Does anyone know what that means? Can someone tell me what paracrine means?
Paracrine is when it is able to act on other cells around it, and autocrine is when it is acting on itself.
Right. So, cytokine action, if you think about that, it either acts on itself or cells right around it, is that the spread is very limited, right? So you need it to kind of propagate. Cytokines are very large hydrophilic molecules with low diffusion, so they really don't travel that well. Think about how much tissue is in the brain, right? It's a very tight structure. There's not a lot of room for things to be floating around in the parenchyma. So in order for it really to propagate a signal, you have to kind of have it going from kind of jumping from cell to cell within a localized area. And that's why we see this propagation by the parenchymal microglia and astrocytes. So how do we shut this off? I think we have a number of lectures on this, but I did mention the IL-1 receptor antagonist. And this is kind of a major way that we see shutoffs of some of these inflammatory signals. So if we think about this for a second, and we think about the NF-kappa B pathway, and I told you that IL-1 is being produced, and so is IL-1 receptor antagonist. And for this purposes, it doesn't really look like this, guys, but just for my drawing, is this receptor antagonist blocks the binding site of the IL-1 receptor, so that IL-1 can't bind. So as you're producing, the same one-to-one expression of IL-1 receptor antagonist as you are the IL-1, you're kind of making sure that you have kind of an automatic shutoff. Other things that help shut off these inflammatory signals include anti-inflammatory cytokines. Within the brain, you'll hear a lot about TGF-beta, IL-10, IL-4, and IL-13. IL-4 and IL-13 are interesting in that they're mostly kind of lymphocyte cytokines, T cell cytokines. So Like work by Yoni Kipnis and others show this sort of feedback on this system by T cells. Turns out some neutrophils and some myeloid cells can make a little bit of IL-4 and IL-13 as well. Glucocorticoids, something we'll talk more about coming up here. This is a hormone that's going to help shut off that signal. Other neuropeptides that are anti-inflammatory are vasopressin, alpha-maltoprin, growth factors. So ones that we've seen over the years kind of been talked about is brain-derived neurotrophic factor and insulin-like growth factor. Both growth factor pathways seem to shut down inflammation. And then we have neurons with contact inhibition. We've talked about this a little bit before. Fractalkine, which is that C3XR1, and then the CD22, CD200. CD200 would be on the neurons. CD22 would be on the microglia. And again, we'll talk more about this. I have a whole lecture on this regulation. This is just from the very first lecture, just reminding you that we do have a number of positive aspects of inflammation. This would be what we talked about last week and part of today, this idea of transmitting these transient immune signals to the brain using IL-1 signaling and other inflammatory cytokines to reorganize the host priorities and have it beneficial on the host organism. And we have all these regulatory pathways in check. And again, this isn't going to be the last time we hear about it, but I think we worry about really exaggerated levels of inflammation and prolonged levels of inflammation. So the pathways really don't change. It's just the level of exposure that does. So you'll see some of the same things, nitric oxide, reactive oxygen species, inflammatory cytokines. Instead of modulating behavior here, now we see delirium, depression, cognitive decline. So these things now are kind of having a maladaptive response within the brain. And again, I think it's the duration of expression. And this just shows that idea that our brain can handle this level of immune-brain communication. It's beneficial to the host organism. We give our innate immune system here, gives us time to form that adaptive response. We clear the infection, and inflammation goes down, and we recover. In some situations, we see that this is amplified. An example of this would be age, where we see higher sort of innate immune responses. It takes a little bit longer has higher inflammation that takes longer to resolve. And then I think when we're talking about conditions, chronic or inflammatory or neurodegenerative conditions, you see this amplified and unresolved. So something to think about as you guys read more about these type of things. Okay, so now we want to go kind of the opposite way and think about signals that come from the brain and influence the immune system. So I want to talk a little bit about the autonomic nervous system. I'll talk about some of the responses, behavioral responses to this. The two major pathways, and you guys might as well write this down now because these are important, would be the HPA and SAM axes. We'll talk a little bit about regulation and then the pathology of a dysregulated response. So this is a very simplistic drawing of the sympathetic versus the parasympathetic nervous system. It's a balance here. So the sympathetic is the fight or flight response, where let's see. Okay, so it's when the sympathetic is going to be the fight or flight response. It's something you've probably heard about, somewhere in your biology courses, where it's the increased heart rate. It's that mobilization of energy, where you're either going to fight off that rabid dog or you're going to run away from it, right? And this is all going to be coming from norepinephrine from the brainstem. So you're going to see this from the spinal cord, and you're going to have this norepinephrine signaling within organs, right? The counterbalance of this is that the organs can signal back to the brain through things like the vagus, right? We've talked about this. And this is acetylcholine based. And this is kind of the rest and digest pathway. So it is too simplistic, and any sort of neuroendocrinologist, and if any of you guys are out there, will know that this is way too simplistic, but it does set up a system counterbalance. So there are some sensing signals that come from organs and use this sympathetic pathway, and there are some outflows from the brainstem using the parasympathetic. But really, the strength in the signals really go in opposite directions. Does that make sense? So the sympathetic is sort of the outflow, and the parasympathetic is sort of the inflow. So this is probably some too basic of neuroscience for you guys, but if we look at the two parts of the nervous or parts of the nervous system here, somatic nervous system, basically you see the cell body. We have our myelinated axon. This is a motor neuron. This is going to be a direct. So these are very long axons, right? That then synapse on skeletal muscle. This is going to be, a motor neuron. Where the autonomic nervous system looks a little differently, we have these preganglionic fibers that synapse on a autonomic ganglion, and then we have these neurons then synapse on smooth muscle, glands, and cardiac muscle. So these are all things that you don't have to really think about in terms of their regulation. Some differences with the autonomic nervous system: there's no specialized postsynaptic sites. They're highly branched. Postganglionic fibers, pre and post, are about 1 to 10. And then the parasympathetic is about 1 to 3. And the neurotransmitters use diffusion to reach targets. So this is sort of the difference between the sympathetic and parasympathetic. Again, this would be a sagittal brain section through the brain and spinal cord. And what we see here with the sympathetic nervous system is what's called the sympathetic chain. So this comes off of the spinal cord. So all the ganglion here are within the spinal cord, and then they go out to, outflow to all the major organs here. You see going to the heart, lungs, spleen, stomach, adrenal gland, bladder, reproductive organs, right? So I know some of you, if you're in the Phil Popovich lab or Dynamic Tide lab, you guys are interested in how spinal cord injury kind of affects this sympathetic reflex. You can have a lot of patients with spinal cord injury have many effects aside from just paralysis. So I don't know if when Phil comes in, if he'll talk about that or not, but it's definitely an interesting area of study. And then the parasympathetic is basically receiving signals from all these same organs, and it's going into different cranial nerves. Anyone know what cranial nerve X is?
Vagus nerve.
That's right. So yeah, this is the vagus right here. The vagus that we've talked about as kind of being important for interpreting a number of the peripheral signals that start in the periphery and go to the brain. Okay, so sympathetic versus parasympathetic. Now there's 3 divisions of the autonomic nervous system: the enteric, the sympathetic, and parasympathetic. These are considered visceral sensory systems, so they're involuntary, like you have really no control of these systems. You can be controlling smooth muscle, cardiac muscle, and endocrine glands. The sympathetic is the preganglion neurons are clustered in ganglia in the sympathetic chain along the spinal cord. I showed you that. That's that fight or flight response. Parasympathetic, the preganglion neurons are in the brainstem, outflow through cranial nerves. Parasympathetic is going to be that rest and digest, maintain heart rate, respiration, metabolism, sort of this homeostasis. Now, why am I going through these different systems, and why is it related to immunology? Well, the sympathetic nervous system is hardwired into the lymph nodes, spleen, and bone marrow. So now we have a direct connection from the brain to very important immune organs. And you ask yourself, well, why does this happen? So what we see is that from the CNS, using norepinephrine signaling, you guys know what norepinephrine is? The other word for it?
Noradrenaline.
What's that?
Noradrenaline.
Yes. So it's basically adrenaline, right? So this is that adrenaline pathway beta adrenergic receptor signaling. So we see these signals innervate into the bone marrow, the thymus, the lymph node, the spleen, and the gut-associated lymphoid tissue. So it's going to be released in these tissues, and there's no direct synapse. So the neurotransmitters diffuse through there, and guess what? Immune cells have receptors for norepinephrine. They're called beta 2 adrenergic receptors. So all these cells have this direct connection to the CNS and can respond directly to these neurotransmitters. Now, there's also some pathways that feed back to the brain using the sympathetic nervous system. These have been identified within sort of the bone marrow and with lymph nodes. So there is some kind of communication back through these systems. So the parasympathetic uses acetylcholine. There is really no input from the brain to immune organs. So this isn't going to be a major pathway for information flowing from the CNS to peripheral organs. But immune cells do have receptors for acetylcholine. They're called nicotinic receptors, and it will cause a functional change. So we do see that they can respond to neurotransmitters from the parasympathetic. It's just that the organs themselves really don't have any inputs from the CNS that are parasympathetic. These signals obviously go back to the brain, but don't really come from the brain, if that makes sense to people. So we've talked about the three facets of neuroimmunology. So today we're really focusing on number 3 here. So signals that come from the brain and influence the immune system. So really the two major pathways, if I haven't hit you over the head with a hammer already, would be the sympathetic system here, but also the HPA, this hypothalamic pituitary adrenal gland axis. And we're going to see these systems turned on with immune challenges. So it's a way to kind of have a negative feedback response. And we're also going to see them with psychological stress. Okay, so how are we relaying these efferent signals? And what's the function of the communication? You guys have seen this. This is that same idea of looking at feedback. So today we'll focus on this hardwiring of the sympathetic nervous system and sort of feedback with glucocorticoids and catecholamines on the immune system. Again, just to remind you and to hit you on the head one more time, is that the whole reason that these neuroimmune connections work is because cells of the CNS have receptors for things produced by the immune system and vice versa, right? So we know that we've talked about this before, that neurons have IL-1 receptors or cytokine receptors. They can respond signals from the immune system. Microglia astrocytes also have cytokine receptors, complement receptors, chemokine receptors, and all respond to things of the immune system. Same is true going the other direction, right? So cells of the immune system can respond to things from the parasympathetic, from peripheral nerve, and from the sympathetic. So parasympathetic in general is going to use acetylcholine. The peripheral nerve, and we'll see this with some types of damage, or where you see substance P or other neuropeptides are released. These are going to be important for pain. These are going to activate immune cells. The sympathetic is going to use a catecholamine called norepinephrine, which we talked about is also adrenaline or noradrenaline, and then neuropeptide Y. And then we have the HPA axis, which releases cortisol or glucocorticoids. And so the receptors for these are on immune cells, and we have the beta adrenergic receptor on all these cells, the nicotinic cholinergic receptor on all the cells, and the glucocorticoid receptor on all these cells. And why did I draw the glucocorticoid receptor inside the cell? You guys see the difference see, these are on the surface, right? Glucocorticoid is not.
They're cytosolic receptors that translocate to the nucleus when activated.
Yep, absolutely right. Okay, so know this slide. This is, I think, something that's important to kind of differentiate these two pathways. So these are going to be activated in parallel, and you're going to see them activated in the context of peripheral infection. So let's start with the HPA. I know we've mentioned it a lot. The PVN of the hypothalamus is going to release the corticotropin releasing hormone. I think we actually saw that in one of the papers from 1997. It's going to, CRH is going to go to the anterior pituitary, where we're going to see it produce ACTH, or adrenal corticotropin hormone. This is actually released. So you're not going to, this is no longer in the brain. This goes out and it goes to the adrenal cortex, which is located where?
On the kidneys.
Yeah, right above the kidneys. And we're going to see this glucocorticoid or cortisol produced. All right, so this is going to be that HPA release of glucocorticoids. The other pathway we see is the sympathetic adrenal medullary axis, okay, the SAM. And this is going to release both epinephrine and norepinephrine. So epinephrine, the other word for it is adrenaline. Comes from 80% is from the medulla of the adrenal gland. So epinephrine is just dumped into the bloodstream, where norepinephrine is going to be released mostly at the nerve terminals. So 20% is actually released into the bloodstream, but 80% is all going to be at the nerve terminals. You guys get the difference? Good. So we'll take a look here at the outflow of these pathways in terms of what's happening, just to give you a little bit of a visual diagram. And again, these are my little stick drawings, so hopefully you can see them. This is the sort of sympathetic, the SAM axis here. We see it hardwired into the immune system, into the secondary immune organs. And we see it going to the adrenal gland. We're also seeing the release of or activation to HPA with CRH and ACTH going to the adrenal gland, which someone told us is right on top of the kidneys. And we're going to see these increase in glucocorticoids and increase in catecholamines. So what are these things doing? So like the title tells you, glucocorticoids are important for glucose metabolism. So they're going to be involved in energy balance. The other thing that happens is that catecholamines, as part of the fight or flight response, it's going to be increased heart rate and increased muscle tone. And one of the things that it does in terms of what these things do to the immune system, we talked a bit about the Th1 response versus the Th2. If you remember, the Th1 response was more of that monocyte macrophage-driven response, much more pro-inflammatory, versus the Th2. So it's gonna help shift the balance away from a very inflammatory Th1 response, more towards a very specific, less inflammatory Th2, which is going to involve more of that antibody production by B cells. So very important immunomodulatory role. Also, glucocorticoids feedback on the brain too. So this idea of this hard wiring into immune organs was discovered a long time ago. And this actually led to MacArthur Genius Prize. And what they found here was that this hard wiring, they could see these sympathetic fibers going directly into a lymphoid organ here. So you could actually see the tracks, and they could see the signals coming in. And at the time, it was totally unbelievable that you saw this hard wiring. And this is kind of what it looks like. This is another one of my stick drawings, but it shows you something interesting. Again, you have this signal coming in. This is going to be a lymph node where the majority of your immune cells, your sort of surveying immune cells are. And you find that there's a distribution of cells within the lymph node. And what's interesting is that the lymph node here, there are certain cells very, very close to the sympathetic fibers. So it turns out that T cells are very, very close proximity to these, followed by the B cells, and then the macrophages. So you see that if you're going to release something that comes in by diffusion, that the T cells are going to get bathed in it first, and then the B cells, and then the macrophages. And they actually have this sort of inverse proportional relationship of the receptors. So the ones like the T cells very close to the terminals have a lot less receptors, presumably because they're being doused with it. They don't need a lot of receptors to respond. But in order to get to the macrophages within the lymph node, you have to have they can respond to very low levels of norepinephrine because they have a lot of receptors. So it's sort of an interesting biological structure of the lymph node in the context of where the sympathetic intervals or integrations are coming in. Okay, so what does this hardwiring indicate? So it means that the immune system is directly modulated by these neurotransmitters and neuropeptides. So what can happen if we have these signaling pathways going on? We can see that it affects their development and maturation. We see that sympathetic nervous system affects antigen processing, antigen presentation. It can modulate the cytokine profiles. Going to see much more. It's going to reduce inflammation. There's Ginny Sanders is retired now, but she for a long time was a neuroimmunologist here on campus, and what she studied was this beta-2 adrenergic receptors on B cells and the impact on norepinephrine on B cell activation. So what you could see is that you would see a change in the B cell where you have norepinephrine present. You would switch the B cell from making an IgG antibody to an IgE antibody. And this was important in context of allergic asthma, where stress and increase in norepinephrine would increase this process, drive IgG, and you'd see worse allergies. So an example of how, neuroimmunology sort of crosses biology here, or immune biology. Okay, so things to remember. Why is this form of communication relevant to immunity? So it's part of normal immunity. It's evolved, and it seems to influence the B and T cell development. It's important for feedback. So these efferent immune signalings attenuate immune activation as part of a negative feedback loop. Remember from the very first course, or very first lecture I gave you guys, that that the system is set up by a number of positive and negative feedback loops because the cells can communicate on a cell level, right? This efferent immune signaling can help shift a Th1 response towards a Th2. So that's that shift away from inflammation, more towards the adaptive B cell antibody production. So the other thing to keep in mind, and I think for a lot of you who I know what you study in your labs, CNS interpreted signals like fear, threat, and stress can be relayed to immune cells by these same pathways, and they can modulate immune responses. So now you're taking a response that is, interpreted within the brain. It's feeding out to the immune system and influencing the pathway. So the vagus afferents that has this cholinergic anti-inflammatory pathway associated with it. So the vagus will stimulate the release of acetylcholine, and we do see these acetylcholine receptors on immune cells, and it is very, very anti-inflammatory. So if you were to add acetylcholine to leukocytes, you'd see it inhibit migration. If you were to add it to activated leukocytes, you'd see that inflammatory cytokines would decrease. And we know that if you have knockouts of these different pathways, you'd see increased inflammation, and we'd see if you enhance these things, alpha-7 agonist, that you would increase, you see more immunosuppression. So there is this hypothesis that acetylcholine is anti-inflammatory, and it makes sense. The one thing just to kind of point out, because there are people who have made quite a career of studying this pathway, there is no parasympathetic outflow to primary or secondary lymphoid tissues. So there's also no vesicular acetylcholine transporter in lymphoid organs. So there's really no direct signal, even though cells can respond to it, they don't. And so one of the sort of solutions to this idea is that experiments show that this pathway might really be dependent on the sympathetic nervous system. And this is why that ultimately you have these adrenal medulla preganglion fibers that use acetylcholine here, and then actually synapse on the autonomic ganglion that produce norepinephrine. So this kind of solves both issues, if you will, in that acetylcholine is involved in some of these responses, but it actually goes through the sympathetic indirect signaling pathway. If we take a look at some of these effects, and let's focus here on HPA axis again, which is going to release glucocorticoids or corticosteroids. Have you guys ever heard of those before? And you guys watch sports? And you talk about like a football player getting a cortisone shot. Why do they get those?
Usually if they have some kind of like inflamed joint like issue.
Right. It reduces two things, right? It's pain and swelling, right? So glucocorticoids and corticosteroids tend to be very anti-inflammatory. And it sort of sets up this negative feedback loop where glucocorticoids will reduce inflammation, cytokine production, they will reduce leukocyte migration, reduce T cell development, and actually reduce the cytokine receptors on cells. So these are immunomodulators that act to attenuate pro-inflammatory immune events and can enhance adaptive immune responses. Again, that's part of that Th1 to Th2 shift. And this is what I mean. This is the Th1 versus Th2 response. If you haven't seen this before, or haven't thought about this, these are sort of the two main helper T responses. I've told you before that Th1 is a cell-mediated. It's going to be going after things like bacterial infections, intercellular pathogens, and you're going to be driving this Th1 response, which is going to involve a lot of interferon gamma, TNF alpha. And what we see is that we prefer to shut this pathway off because it can be highly inflammatory, and we move towards a Th2. And so glucocorticoids actually do this. They lower the impact of this system and drive this system here to get more of a Th2, to produce more humoral immunity, more B cell production. And this is going to be important to ultimately clearing that infection. And I know I've shown you those graphs before. So to bring some of these concepts together for you in terms of remember, everything we're talking about is NF-kappa B to keep it simple, right? So again, please know this pathway. So how do glucocorticoids modulate inflammation? Well, it turns out glucocorticoids, as someone said, I think it was Kayla, said that they're cytosolic receptors. So glucocorticoids are inside the cell. And so glucocorticoids are lipid signal molecules, so they can go right through the cell membrane. They bind to the glucocorticoid receptor, which is sequestered here to the cytoplasm by heat shock protein. Once it binds, it allows it to, the heat shock protein will be lost, and then they'll dimerize. So you have two receptors together that have the glucocorticoid bound to it. This will again allow a nuclear localization signal. This will get into the nucleus. And it turns out that the position of where these glucocorticoid response elements are actually right next to the NF-kappa B response elements. And this glucocorticoid response sort of cluster here, when it sits down on here, it actually can bump off the NF-kappa B. So glucocorticoids have a lot of effect on cells. One sort of negative feedback loop that maybe you haven't heard about or you didn't know is that glucocorticoids also will kill immune cells, and they'll do it through an apoptotic pathway. So sometimes, the brain will tell the immune system, you guys have had enough fun, it's time for you to go away. And so high glucocorticoids will kill macrophages, dendritic cells, and T cells. So you find that this is a way that you can, not only attenuate immune responses, but also eliminate them. Okay. So this is some classic work done by Serge Rivet, who is at Laval University. He's done a lot of immune signaling work over the years. And what they're doing here is they're giving peripheral LPS injections to and showing increased toll reactivity in the brain. So this is going to be within the supraventricular organs, choroid plexus, and hypothalamus. This is in C2 hybridization for toll2, so you see the expression. You can also give this injection by ICV. So we're seeing this increased toll-like receptor response in the brain. And what they're really doing here is looking to see what happens when you block the glucocorticoid pathway. So in these experiments, Sergey and his team here are giving interparenchymal injections of lipopolysaccharide, which they know they respond to by toll signaling. What they're seeing is that they're giving this glucocorticoid inhibitor, RU486, which some of you may have heard about in some other contexts. But we find that if you block glucocorticoid feedback on the brain, we see increased demyelination. So demyelination here is in white, and necrosis. We're seeing loss of cells here, cells degenerating, dying here. We're seeing this is a labeling for DNA fragmentation. I think it's what's called a tunnel stain. And these are caspase-3 positive cells. So what we're seeing is that as part of a normal immune response, glucocorticoids feedback also on the brain and reduce microglia activation. If you lose that regulation, we're seeing things that we normally don't see in these responses. We're seeing demyelination, we're seeing cell death, we're seeing apoptosis, and we're seeing less live cells. So one thing to note with all this is that you also will read papers that high levels of glucocorticoids cause cell death, which I showed you in a couple slides ago, right? That high doses of glucocorticoids can kill cells. But in this case, in terms of just a normal level of glucocorticoids, is protective against excessive inflammation. Okay. So these are glucocorticoid receptor deficient rats. And what they've done here is given that same interparenchymal lipopolysaccharide injection. And here they're looking to see if they can reverse some of the effects. So what is the main mechanism by which the inflammation is being perpetuated? And here they're using a number of antibodies. And so they're using antibodies against IL-1, TNF-alpha. And basically what they're seeing is that there's a reduced level of inflammation, meaning that the lipopolysaccharide is using these pathways to induce inflammation. If the rats don't have the glucocorticoid receptor, you can circumvent that deficiency by blocking cytokines. So normally glucocorticoids are important for down-regulating some of these cytokine signals. If glucocorticoids aren't present, you can kind of circumvent that problem by adding these antibodies. So glucocorticoids are really critical for shutting off the brain inflammatory signals mediated by IL-1 and TNF-alpha. And again, this is from Sergey's group in 2003, which is a long time ago, but really sets the table for how glucocorticoids feed back on the brain. And it turns out really all of these pathways, the glucocorticoids, the catecholamines, acetylcholine, all suppress NF-kappa B activation. Again, these slides are 20 years old, but really our inflammatory signaling pathways are still NF-kappa B based. And they can do a number of different ways. It can be knocking off the promoter site, like I showed you. The acetylcholine receptor blocks NF-kappa B activation. So does the beta adrenergic receptor. So ultimately, all these things sort of fall on this one target. So one thing to think about as we start to think about the different immune pathways that come into the brain, we talked about this afferent signaling of peripheral immune activation, or IL-1 mediated, going into the brain. And then we see that there's the output, right? So the brain interprets these signals. There's some level of inflammation in the brain, and then the output is the activation of the HPA and the SAM. This is going to involve glucocorticoids and catecholamines. This is going to be showing us this negative feedback or immune modulation. And in this instance, what we're doing is basically two things. We're hoping to move the immune system away from that Th1 inflammatory response to more of a cell-specific antibody Th2 response. And the second thing it's doing is helping to reduce microglia reactivity in the brain. So two important functions of what this pathway is doing. So do we need an immune activation to drive these same pathways? And I told you guys, the answer is no. So we can actually get these same pathways activated with fear, threat, stress, and my little dots here of where the stress inputs are going in are not accurate. So they're just kind of going randomly in the brain, but they are, all these things will be interpreted in different parts of the brain, but my drawing here doesn't show you where that's happening. Just know that they're being interpreted. Okay, so these signals go into the brain, and then the output can be variable. Okay, so you're going to see that the output in these situations can be HPA and SAM activation with glucocorticoids and catecholamines. And here's the confusing part: you can see either enhanced immunity or impaired immunity. All right, so it's going to be very context dependent on what happens here. So if we go back to the fight or flight response, a little bit of a high school biology type of intro into the sympathetic nervous system, is you're going to see that one of the things it does for the immune system is it enhances leukocyte trafficking and cell-mediated immunity. And this is an example of enhancing immunity that's thought to be beneficial. So I don't know if I show it. Next slide. Yeah. So think about, the example of, going back to probably the way your high school biology teacher taught you about it. And the one I remember, the one I used, is because it was the one told to me, was that, if you're cornered, by a rabid dog, right? And you get that fight or flight response, and you have that adrenaline running through your veins, and your heart's beating fast, and your body's breaking down muscles so that you can move quickly. What it's also doing is it's releasing immune cells. So one of the things that get released are neutrophils, and neutrophils will travel to the skin. And the idea is that if that rabid dog bites you, you have all these neutrophils there to respond to that and potentially clean up any infection or anything that might occur with that bite. So it's thought to be a beneficial connection between the fight or flight and the immune system. The enhanced leukocyte trafficking, enhanced Th1 responses give you that little added protection. So this slide is just showing you some of the enhanced effects of what this pathway would do in terms of increasing Th1 processing, increasing chemokines, interferon gamma responses, and monocyte release and stimulation in the bone marrow. So you're driving up immune cell, not only driving the release of them into the bloodstream, you're also increasing them in the bone marrow. And some of the work that I've done with Dr. Sheridan on campus really shows that you could put this system in overdrive with particularly a chronic stressor. So it looks like, for the most part of our stress pathways, such as fear, threat pathways, are relieved immune cells and modulate immune responses. And for the most part, this is thought to be a beneficial response. And then the question that many of you think, or maybe are thinking, is that, why do we hear so much about stress and immune dysfunction? Well, I think it's because of the duration and course of stress. And this is the big sort of context dependent changes. So chronic stress, when you're thinking about long-term or stressful situations that occur, not just the acute stress of having to lecture in the morning, right? But something in your life that is causing chronic stress, like marriage problems or things like that, or worrying about money. That's an impact. This is an impact that's driving the system every day, all the time, on your immune system. So there are a lot of examples of how this input, this chronic stressor, can drive these same pathways, the HPA and the sympathetic nervous system, to impair immunity. And there's a lot of different proposed mechanisms of how this happens. So these are ones that we see. Okay. I should be able to finish on time for those of you worried. These would be more chronic stressors. Academic stress would be one of them. Marital discord. One of the big ones studied here at Ohio State campus is this primary caregiver of a demented spouse or a sick child, child with cancer. This, for the most part, this has a negative effect on the immune system, where we see things like in the innate capacity, we see reduction in natural killer cells. So this has come to the link of sort of immune dysfunction. And also, if you lose natural killer cell activity, you actually lose some cancer, looking for cancer cells to eliminate as part of the innate immune system. So there's this link between stress and cancer. We also see that stress affects wound repair negatively. It can increase circulating cytokines. We see this reactivation of latent viruses. And we see, stress affecting the antibody production, something that's actually quite relevant in today's times with, high stress individuals not getting good antibody responses to the COVID vaccination. And you have these sort of situations, you're also going to increase the number or maybe the severity of infectious episodes. So we see that sort of stress has the effect, chronic stress has the effect on infections, in that we see more clinical symptoms. We see reduced recovery from infection. We see stressed individuals have a prolonged recovery from surgical procedures. We can see this idea of having cognitive and behavioral deficits with stress. And this sort of stress and cancer link is that these things can also promote tumor growth and progression. So a lot of interactions here driving these pathways. This is just some work I wanted to highlight that was done here at OSU by several famous scientists. Started by this group by Ron Glaser, who's unfortunately no longer with us. His wife, Janice Kiko Glaser, is still here. She's the director of the IBMR now. And John Sheridan, many of you probably know him, a neuroimmunology stress researcher. And so they actually got together and to study this psychological stress in the context of a caregiver for a demented spouse. They're looking at seasonal flu vaccines. And they found this sort of neuroimmune interaction of this. And this is sort of of what's needed to happen. So when you get vaccinated, you need a Th2 response. This is that here. So this is the flu particle here, or the antigen here being expressed on MHC class 2. This is your helper Th2 cell. This is going to use IL-4 to drive B cell production of antibodies here. So this is actually going to turn them into a plasma cell. So they're going to be producing antibodies specific to that antigen. This is going to give you protection against flu viruses. And high glucocorticoids can disrupt this pathway. And what they found was that the high glucocorticoids affected this pathway. So you actually had high stress individuals were producing, you could have less antibody protection. So not only was there less antibody, but the people weren't getting very much protection. They were still getting the seasonal flu. So this is a kind of a landmark study showing this connection between stress and immune cell function. They also did another classic study done here on campus. This was the psychological stress of med students studying for the board exam. And they compared these individuals to medical students who, weren't taking their boards and actually were like at Lake Havasu for their spring break. And what they found was that the high stress individuals studying for the board exams had a reactivation of latent herpes virus. This is something that Dr. Glaser was an expert in. And part of it was this Th1, Th2 response idea that I've shown you multiple times today. So in order to keep a latent virus in check, and all herpes viruses are all endemic in human populations, but we need our immune system to really to keep their viral production and replication in check. And so what happens is in high glucocorticoids, with stress, you start to shift away from the Th1 response as we want to normally. But in this case, the sort of unintended consequence is that now, as you start to do this, now we see that there's reactivation of virus. So there's two types of latent herpes viruses that they studied. Herpes simplex, that's the one that's going to give you the cold sores, and then Epstein-Barr virus, which causes mono. Some of you probably even might know individuals, high stress individuals who seem to have gotten mono at some point in their lives. So the Th1 immune response controls the pathophysiology of both the lytic and latent phases. The lytic phase is when the virus starts to replicate and use cell machinery to make more viral particles. So we see that stress promotes latent Epstein-Barr virus reactivation. So we see less immune control of this, less T cell memory responses, and less Th1 associated cytokines. One interesting thing just to throw in there is they also did this, someone else did this study in West Point cadets. And so West Point, they have two types of exams. They have, their big, midterms and finals and things, but they also have physical exams. And what they found was that they saw the same effects in sort of the studying for the board exams at West Point, but the physical stressors did not activate these same pathways. So basically the take-home message here is that stress induced a shift in cytokine profile away from Th1 towards the Th2, and the virus was reactivated. And this is just kind of showing you that you need this kind of constant Th1 response against infected cells to keep them in check. With stress, we saw this change and more. And what they found here is also related to the NF-kappa B pathway, in that they found that the glucocorticoids were knocking off NF-kappa B off the promoter site. So this is why you're kind of seeing that shift away from Th1 towards the Th2, but in the context of viral reactivation, this increased viruses spread or replication. Yep. So that is it for today, and I think I'm dead on time. That's great. Let's see, and we'll keep recording here for a minute just to see, does anybody have any questions?
I actually have a question just about the recording for last week. I couldn't find it on Carmen. I don't know if it's up and I just missed it or.
Okay, so the answer to that question is that it's not there. I'm having trouble loading it on Carmen. I do not know why. So we talked a little bit in the beginning of class about maybe me putting on my YouTube channel or sharing it with you through a box, like a box shared file. If you want it and you can't, if you need that file, or somebody in the class needs that file, have them contact me. I'll try to figure out how to get it to you. I've tried multiple times now to get it on there. I can load other ones on there, but for some reason, not that one. So
that one, just, I know at least Jonathan and I were at a review session, so we missed it, but so okay. Maybe just that one.
Yeah, we'll figure out how to get it to you. And same with anything that you guys have missed and I can help you with, let me know. I want to make sure. I don't know why Carmen's being unfriendly with that one, but it works. I watched some of it. Look, I mean, I think I look pretty good and sound pretty good in it. I don't know. Just doesn't like that one. It's not any bigger than any other file, so. Yep. Anybody else?
Yeah, I have just more of a broad question. So when it comes to glucocorticoid binding to, their sequences in the DNA similar to or nearby to the NF-kappa B binding sites, and interfering with the transcription factor binding there, do you think that, so like all of that work, is that specifically in immune cells? So like those examples you gave, like that's in immune cells, but do you think that is conserved and like a same mechanism happening in like astrocytes or other types of cells, or do you think that could be a context dependent, event going on to like that happens in immune cells, but like maybe not in like other types of cells?
That's a very interesting question. I would say that the latter is probably true, that it's probably very specific to immune cells. I mean, I don't know how I think astrocytes are very important cells and very interesting cells, but from what I've seen, I don't think astrocytes have the same machinery, right? So what glucocorticoids may be doing in those cells might be a lot different than what they're doing in immune cells. And Isabel's absolutely right. All the context I showed you today would be what's happening in immune cells. So that NF-kappa B signaling pathway and those glucocorticoid interactions would be relevant to immune cells and not, other cells of the body. And in my view, my undergrad is in cell and structural biology. So I grew up studying how cells interact. And so every specialized cell has a whole different complement of signaling systems. Otherwise, every single component out there, whether it's insulin or growth factor, we're all doing the same goddamn thing in all the cells. And that's not what happens, right? So each cell is specialized to respond accordingly or not respond, right? So, a lot of these cells don't respond to insulin because it makes no sense for them to do that. So that's sort of what I think. So I think the latter, Isabella. Okay. Anything else?